Good day. In this video, I present to you a latest research work done at the Machine Vision and Intelligence Systems Lab. This work proposes a multi-stage framework for automated sphelometric landmark detection addressing resolution and spatial invariance. Starting with the introduction, sphelometrics is basically a branch of dentistry and medicine, which is commonly used by orthodontists to analyze the relationships in the human skull. It is used to diagnose cases such as malocclusion, which is misaligned teeth, facial abnormalities, and even for aesthetic purposes. Now, the most vital part of sphelometrics is the sphelometric analysis, which is done on sphalograms, which are 2D projections of the human skull. Sphelometric analysis is a three-step process in which firstly landmarks are marked, then angles and distances between these landmarks are found, and then a qualitative assessment of these angles and distances is used for diagnosis. As we can see, the most vital part of this process is landmark detection, which traditionally is done manually, which has a few limitations. Yes, it is time consuming, complex, and open to both intra and inter observer variability. This problem, however, could be solved if we automate this process. That is, develop an automatic system that will take the sphelogram as the input and give the landmark locations as the output. Therefore, the motivation behind this research is to construct a fully automated tracing framework that can localize phonometric landmarks accurately, consistently, and rapidly. For the construction of such systems, datasets play a very important role. The first benchmark dataset was the ISB dataset that contains 400 spectrograms coming from a single imaging device with 19 landmarks marked per spectrogram. Most of the methodologies proposed in literature uses this previous benchmark dataset. However, this is a small dataset with very less variability. This issue was solved by the RS dataset, which was proposed by our lab at the CIFA 29 challenge and is the new benchmark dataset. This contains 1000 spectrograms coming from 7 different imaging devices with 29 landmarks annotated per spectrogram. RS shows great progress due to its large training set and resolution and spatial variability, which was not found in any of the previous datasets. Despite its improvements, there is no methodology to date that caters to the variability found in this dataset. Thus, based on the in-depth study of the datasets and methodology, we found four major research gaps. Most methods use the ISB dataset, and we know ISB lacks generalizability. We cannot comment on the generalizability of any of these methods. Secondly, since ISB lacks resolution and spatial variability, none of the methods cater to it. So, most of the methods in literature train separate networks for each landmark, increasing computational costs. And lastly, since vision transformer based models are the state of the art right now, they although perform better quantitatively, they increase the computational costs when compared to the CNNs. Thus, a comparable generalizable performance using CNNs is necessary. Thus, the problem statement of our research is that the existing methods for automatic sphelometric landmark detection are hindered by the challenges of insufficient generalization across diverse datasets and a lack of robust training data, necessitating the development of a more generalized model that can accurately detect landmarks across various imaging conditions, resolutions, and spatial dimensions while avoiding overfitting and computational inefficiencies. The proposed solution to this problem is to address the lack of generalization in existing sphelometric landmark detection models. We propose a framework trained on the RS dataset that incorporates multi-resolution and spatial dimension invariance while effectively learning local and global features to enhance accuracy and robustness across diverse clinical conditions. Thus, our proposed methodology is a three-stage end-to-end sphelometric landmark detection framework where the first stage regresses the 9-edge landmark locations, which are used to extract the craniofacial region, which is then fed to the second stage that regresses all landmark locations simultaneously, which are used as pseudo landmark locations to extract high resolution crops around all of the landmarks, which are then fed to the refinement stage to learn local features and give the final landmark prediction. This shows the impact of each of the stages qualitatively. The final block specifically highlights how the landmark refinement stage moves the prediction from the detection stage more closer to the ground truth. To look with a deeper lens, the input to the first craniofacial region detection stage is the original spherogram. This stage allows for spatial invariance, and the output is the 9-edge landmark locations, which are then used to extract the craniofacial region based on the minimum and maximum x and y values. The extracted craniofacial region is evaluated using detection accuracy, which is a new evaluation metric proposed in this work. The extracted craniofacial region serves as the input for the Cephalometric landmark detection model, which contains the backbone feature extractor and semantic fusion block for multi-scale feature learning and global feature learning, introducing resolution invariance. The output for this stage are the 29 heat maps, each corresponding to one landmark. These predictions are then scaled back to the original image where keeping the prediction at the center, a 256 cross 256 crop is generated which is both high in resolution and captures the fine structural details. This crop is then used as the input for the refinement stage that allows for local feature learning and is computationally efficient. The output of the framework is the refined landmark location. This module is computationally efficient because it uses a single unified model to refine landmark locations for all landmarks and does this by using the concept of batch processing, where a single batch corresponds to a single sphelogram. The results achieved by our model is as follows, with the MRE of 1.69 and 81.18% of the landmarks lying in the clinically acceptable range of 2 mm. For the further verification of the generalization of our model, we evaluated it on the ISP dataset, where we achieved clinically acceptable performance and comparable results with all other methodologies in literature which were particularly designed for the ISP dataset, proving the generalization capacities of our framework and the RS dataset. Thank you.